Hi all. So maybe the second midterm was a little harder than the first because the average wasn't 90 this time, it was 84. So if you felt like it was a little harder, or if your score was a little lower, you're in good company. And in answer to probably more than one of you noticed that there were a few questions from earlier material because we needed to fill it up to try to get to 100 questions. So that could happen again in the future. But in general, the tests are not comprehensive until we get the final exam, and then that will be. Pay no attention to the man behind the shirt. Excellent. Any questions about the exam? Yeah.
Wanted to look at our chat we filled out from.
Yes.
Set up a meeting with him. I just
email Antonio. Yep. And arrange it. Don't yet, because we still have two more folks that for various reasons haven't taken the exam yet. So postpone. Maybe after the break. Is that okay, Abdur? After the winter break, it's good. Yeah. Give me a couple of days. Or you go ahead and email Antonio, and then I'll give him the go ahead. Good. We have at least two folks that seriously could not take it yet. Format work, all that. Okay. Let me tell you what's coming up. So if you look at Perusal, you'll see I have you slated to read chapter 7, which in the first edition is kind of a hybrid chapter. The first part of it is about apoptosis or cell death, which I'm going to talk about now. The second half is about sexual differentiation. And so you'll also see a new assignment on Perusal, which is a PDF of a draft of the new chapter in the second edition. Okay. So what I want you to do is to read, the instructions are there, but go to the Perusal chapter 7 as it currently stands and read the first several sections that are about apoptosis. Then stop reading that chapter and go to the PDF I just posted, where you'll read about hormones. And then at the end of that, go back to chapter 7, and having learned about hormones in the new chapter, you can now appreciate the sexual differentiation that completes that chapter. Sorry, it's complicated. I've tried to put directions in the various places as you go, but there you are. Questions about that? And it's a PDF with the figures pasted in, but at least they're the finished figures. So at least you don't have to look at my scrawls and try to figure out what I'm trying to write there. Okay? And believe me, you'd never want to look at any figures that I have drawn. That would be, tough. Although I should tell you that for 20 years, I taught at Berkeley 4 courses a year where I never used any slide, only used chalkboard. And one of the things I noticed is that with the advent of PowerPoint, I talk faster. Having to take time to draw those things on the chalkboard meant that I necessarily had to slow down. And so it's a mixed bag. Yeah. Okay. Questions about the course? All right. Let's talk about apoptosis, naturally occurring cell death, which is an important part of every development of every organism, every metazoan that I know of, including C. elegans. Remember in C. elegans, sometimes when we were tracing that mitotic lineage, there'd be some cases where there'd be a final division and one of the cells would take on a task and its sister cell would die. And so probably and as we're going to see, many of the molecular mechanisms behind that death in C. elegans, they have total homologs in us. So somewhere back before our common ancestor with C. elegans, natural selection came up with a system of sometimes making more cells than the organism actually needs. We can speculate later about why that might happen. And the basic mechanisms behind that have been the same. The first thing I'll tell you about is that word apoptosis. So it was coined by some cancer researchers in the early 60s, as I recall, And you can read the article for yourself. It's cited in the book. And they quite clearly point out that that second P is supposed to be silent, as in other. It's a Greek-derived word, as in other words like that. And yet you will hear people talk about, I just came back from neuroscience, you will hear people say apoptosis. Okay, so just go with it. But I'm telling you, the guys who coined the term, they thought it should be apoptosis. And we're going to learn about it. And one of the things that's most interesting about apoptosis is that it took a long while. It wasn't until, the second half of the 20th century that people could accept it was there, even though there was lots of indications of it all along. Because it seems counterproductive. Why make a cell if you're not going to use it? And so there's some places where it's pretty obvious why that might be a good idea. It turns out there was a time when your hand was getting made that, yes, you had fingers and there was a web between the fingers, right? And for whatever reason, it seems to be easier in development to make fingers first as part of a sheet of tissue, have the bones form in various places within that sheet, and then for the cells between the various bones to die away. And you can see that quite deliberately if you compare chickens and ducks. So both chickens and ducks early in development have that same sort of arrangement that you had once upon a time with here are the digits and here's the webbing between them. In ducks, they stay. But in chickens, before hatching, there's a period where, see all these dark cells there? They are marking cells that are dying. So the way the chick ends up with individual toes that are not connected by webbing is because those cells died at the right place and in the right time. So sometimes what apoptosis seems to be doing is simply sculpting tissue to end up making a final body that works. And the first folk to actually get a handle on cell death in animals, there was growing indications that cell death sometimes happens in plants during development, but people working in animals had a hard time. It never occurred to them. And one story can kind of get that across. There was a German researcher, Viktor Hamburger, who was working with Spemann. Remember Spemann of the organizers in Freiburg. And he's a graduate student there. And he was studying chick development. And it was Hamburger who would eventually come up with a system of how to look at a chick embryo you just exposed. And he came up with a numbered stage to let you know at what stage of development it was. And you needed that instead of how many days ago did the hen lay the egg, because depending on temperatures and things like that, right, some eggs hatch faster than others. And so he came up with a way of standardizing the stage of chick development that's still used today. And he was interested in the spinal cord and in motor neurons. So he noticed something, that he could do experiments where he could remove a limb bud in a chick embryo early enough that the animal would survive, but late enough that it wouldn't regenerate the limb bud, right? So he's getting the right point in development to avoid the regulation he doesn't want. And then he noticed that on the side where the missing limb bud was, a week later, still before the chick is hatched, a week later, he killed the chick, looked at the spinal cord, and he noticed there weren't nearly as many motor neurons on that side as there was on the side where the limb was intact. And he reasoned that it must be then that the periphery was telling the spinal cord how many motor neurons to make. And he assumed that this difference that he saw was because there had been a failure of neurogenesis of motor neurons at that point. Another possibility, the one that turned out to be true, was that this arose because motor neurons that would have been there died. But he didn't even think of that. Or he could do something else where he could add another limb bud there so that you could end up with a three-legged chick where there are two legs on the same side. And what he noticed is in the spinal cord where there were two legs out there were a lot more motor neurons. And so he interpreted that clearly enough. Oh, and if there's bigger periphery, it will induce more neurogenesis and therefore more motor neurons. And that turned out to be quite wrong. So here's Hamburger in those days. It happened to be his good luck that he came to America for a postdoc in St. Louis. And while he was gone, a very famous philosopher, those of you taking philosophy have heard of Martin Heidegger, who was a card-carrying Nazi, an important Nazi in the party. He wrote Hamburger a letter and said, "Oh, you know that faculty position that you had? We've just passed all these laws to keep Jews from having faculty positions, so you're fired." And Hamburger, being Jewish, decided to stay in America where he could work, and of course that may have saved him from the Holocaust. So he'll stay in St. Louis the rest of his life. And he realized that there were 4 different mechanisms to account for any manipulation that changes the number of neurons, right? One is differences in neurogenesis, right? So maybe when you add an extra limb bud there's additional neurogenesis on that side, and that's why you end up with more motor neurons. Another possibility was that maybe the number of neurons that are born is the same, but when you add an extra limb bud, some of the neurons in the spinal cord that would have taken another role change their fate and become motor neurons. So they have altered their differentiation. A third possibility that can also sometimes be hard to pin down is that maybe there's a difference in migration. Maybe when your manipulation causes more neurons to be someplace, it's because neurons from other places moved over there that otherwise would have been someplace else, and so you don't recognize them. Everyone get that? And then the last possibility is that the manipulation changed the number of neurons because it affected the process of apoptosis. To this day, anytime you do a manipulation that changes the number of neurons anywhere, those are the only 4 avenues available to you. Right? There's no other way to do it. And we'll run into it several times in the future. What finally convinced people that there might be some cell death going on is when people noticed, working in frogs, not chicks, they noticed that about the time, first of all, if you simply do a count of how many neurons you see something interesting. So Hamburger counted all the motor neurons in the spinal cord of chicks. And what he noticed is that early in development, about 5 days after incubation, there were 20,000 motor neurons, but by the 10th day of incubation and until the rest of the animal's life, there were only about 13,000. So a whole bunch of motor neurons came up missing. And so that was one indication that, yes, indeed, some motor neurons changed. Now, it's still possible that they didn't die, that they just migrated somewhere else. Or, what's another possibility? They didn't die. They didn't migrate somewhere else. What else might they have done? Good.
They differentiated.
They changed their minds. They were looking like motor neurons, at day 5, but, we're not needed here. So they changed differentiation, by which we mean change gene expression and change their fate. And now they no longer look like motor neurons, and we've got no way of keeping track. The other possibility is
Levi.
Yes. And another possibility is they migrated away. They went somewhere else. And now you're not finding them in the ventral horn of the spinal cord, and so they're missing, right? And this phenomenon that if there's often more neurons in a population early in development than later seems to be almost universal in the nervous system. As long as you can recognize the neurons, you have some way of telling them apart, and you can keep track of them, there's almost always more of them early in development than there are later in development. It would be Nancy Forger, who's now at Georgia State University, who confirmed that the same thing happens in human spinal cord. the 10th week of gestation, there's like 170,000. And before they're born, they're fewer than 125,000. And that seems to be stable the rest of their lives. Or you can not talk about the CNS. You can look in the brainstem nuclei. And here you can see again in chicks that there's 15,000 early in development. And well before they hatch, it's down to 700. Or you can look in the periphery where there's an autonomic ganglia. I forget which one this is, in chicks, where there's a lot more of them early in development than there are when they hatch. So, which of the four was going on? And then people noticed in frogs that just about the time neurons go missing, there's these weird profiles they could see in their stain that they never saw before and would never see afterwards. And they were called Pyknotic profiles, where it looked like suddenly the cell turned into goo, and like the nucleus dissolved into little bubbles and turned dark in Nissl stains like this. And it was these Pyknotic profiles. And the weird thing was that the number of Pyknotic profiles was much smaller than the number of missing neurons, in this case, motor neurons. And so it would be in frogs where people would keep careful track of this. So here we're looking at the number of motor neurons in frogs at various stages of development. And you see there's 4,000 at one stage of development. And by the time they're an adult, there's only, 1,500 as they're counting them. And then they also, in the same tissue, kept track of how many Pyknotic profiles they saw. And early in development, they saw virtually none. And just at that time, when the number of motor neurons is declining fastest, that's when there was a peak of Pyknotic profiles being seen. And then as the number of motor neurons asymptoted and stabilized, Pyknotic profiles disappeared again, so that by the time you looked in adults, you just never saw that. You never see those Pyknotic profiles. And so that was at this sort of data from mid-50s, 1950s, convinced people that the motor neurons weren't de-differentiating, they weren't migrating away, they were dying. How's that so far? Later, when people started watching C. elegans developing, where you can watch each individual cell and keep track of them in those mitotic profiles where mitotic lineages, where there's a final division and one dies, they would watch the other cell undergo the same sort of changes that you see in pycnosis. So they're dying, and it's deliberate. And I'll remind you that in C. elegans, if you kill the cell that would normally take on a function and ask whether its sister cell will change its mind and stick around to pitch in or die, for the most part, the sister cell goes right ahead with this program and dies anyway. How's that? Because notice that the scale here is totally different. The number of degenerations, number of Pyknotic profiles, this is in the 70s. And over here, this scale's in the thousands. And from that, people realized that because the number, and remember, in this sort of tissue, you only get to look once. At the second that the animal died, you get to see how many Pyknotic profiles there are and how many motor neurons there are, right? You don't get to watch it as it's happening, as people would later in C. elegans. And so they realized that once a cell decides to die, it undergoes pycnosis, and it goes from looking like a perfectly healthy cell through a Pyknotic cell, and then it's totally disappeared in about 3 hours. So it's one of those situations where the cell looks perfectly healthy, and it's like you would have no idea that anything's wrong. And then within 3 hours, not only is it dead, but it's been totally dismantled. And presumably the portions of it are recycled to use in other ways. So that means over here, you can calculate that there's about 50 motor neurons dying per hour, right? Or here, about 30 motor neurons dying every hour. So far, so good. And so when you see things like this, it's still an open question which of the four methods are going on. But for the most part, you've got other ways of confirming that indeed this is a matter of the cells dying and getting dismantled and the portions of it getting reused for other parts of the developing cell. About the time that Viktor Hamburger was sheltering in St. Louis while World War II went on, he by the way, was good friends with Raymond's graduate student who did the organizer experiment, Hilda Mangold. He was good friends with her and had kept track of her son Christian after she died. And eventually he learned that Christian died in what Hamburger always called Hitler's War. So while he's sheltering in place in St. Louis, Rita Levi-Montalcini was an Italian scientist who was a physician who had a physician teaching, in Italy. And about the time that Germany passed its laws saying professors couldn't be Jews, Italy started passing its laws saying professors were not allowed to be Jews. So she lost a position at the University of Turin, but she loved her research, so she just kept doing the research at home, looking at chick embryos. She'd never met Viktor Hamburger, but she'd been reading his papers and was very interested in this idea that the periphery was causing what she then thought was more motor neurons getting born, because that's what Hamburger thought. And so she will continue her research through World War II, during some of which she's in hiding because she was a Jew in Mussolini's Italy. And, as she said in her memoir, she continued doing her work with the chick embryos, but it was very important that every embryo was eventually harvested to feed the family because they were starved. And she was also interested in motor neurons, but she noticed that Hamburger's experiments didn't just change how many motor neurons were found on each side when you take off limb buds and add them, but rather that the size of the dorsal root ganglia were also larger when you had two limbs on a side, and they got tiny if there was no limb on the side. And her initial assumption was, so when you have two limbs, there's more DRG cells getting generated through neurogenesis, and that way you end up having the right number of DRG cells to provide sensory information for two legs on that side rather than just one. Everyone get the idea? And how's that? She survives World War II. She and her sister and most of her family, they hid well. She survives World War II, and as soon as it's possible, she wants to go do a postdoc, and who else is she going to do it with in the whole world but Viktor Hamburger in St. Louis? So she goes to St. Louis and will also be there for decades at Washington University in St. Louis. And while studying there, another student of Hamburger's had found a weird thing that you could do with chick embryos, which is the guy was transplanting cancer cells into chick embryos, and he saw that some cancer cells would continue to grow inside the chick embryo. And he's mostly interested in development, but also cancer. But he goes on to do other things, and Levi-Montalcini comes, and Hamburger says, you know what? These tumor cells, you stick them in the chick embryos, they grow, and a weird thing happens. He said, and she looks and confirms that on the side where the tumor is, that the dorsal root ganglia are a lot bigger than on the side where there's no tumor, and that the sympathetic chains are also larger on the sides with the tumor. Also, she noticed these cells, they love to innervate these ectopic tumor cells. And again, her initial assumption is, something in the tumor's releasing some factor that's boosting neurogenesis in the dorsal root ganglia as well as in the motor neurons. And that's kind of cool. So now all we have to do is figure out what the tumor's putting out because it's controlling this process. In the meantime, by the 1950s, it's clear, the motor neurons it isn't influencing motor neurogenesis, it's influencing cell death. And so the reinterpretation is, this tumor cell is putting out something that is preventing cell death, apoptosis, the name wasn't there yet, in the sympathetic chains in the dorsal root ganglia. So let's find this substance to see what it is that's causing, that's keeping cells from dying. Cool, right? And it did not escape their attention that finding a factor that keeps neurons alive might be a useful thing to know. Right? So they start on some experiments to find out what the tumor is making. So I'm telling you the story of nerve growth factor. You cannot be a neuroscientist unless you know the story of nerve growth factor and how it was found. So if you haven't heard this story yet, I'm making you a little closer to becoming a genuine neuroscientist, whatever your major is. So they start looking at the stuff that's being produced here, and Levi-Montalcini comes up with a brilliant assay of how to screen for various substances to see which one might be the magic factor they want from coming from tumor cells. And one of the highlights of the nerve growth factor story is that they were unbelievably lucky. I happen to think I'm one of the luckiest people I know, but in this experiment, they were unbelievably lucky. Number one, the mouse sarcoma line they chose, it turns out there is no other tumor line in the world that produces this stuff. And it was the first one they tried and the only one but their luck gets even better. Hamburger brings in another postdoc, a protein chemist, guy named Stanley Cohen, into the lab there in St. Louis, in Hamburger's lab. And Levi-Montalcini has this assay where she'll take little bits of dorsal root ganglia from a chick embryo that are young, and she figures out how to grow them in drops of water. So they're being suspended in this chamber, growing in a little drop saline. Everyone got that? And now all we have to do is take tumor stuff, the stuff the tumor's secreting, and we'll go through old-fashioned biochemical fractionation to see which fractions make those cells get bigger. And so if the little bit of ganglia, if it was getting some of this stuff, it would send neurites out in every direction. So it's nice and obvious. So another thing that's important in science, in biological science, is number one, we talked about before, ways of screening lots of animals at once. But another secret to succeeding in biological sciences is having a good sensitive assay, one where you can, keep track of a process in a sensitive manner. And so Cohen says, the first question is this stuff, and so they find a fraction from tumor secretions, and it's having this effect, so they figure, the important stuff is there. And Cohen says, the first thing we need to do is see if it's a protein. So let's take some of the fraction. When we add the sarcoma fluid, we get that halo effect. So, it's in there. And now we're going to treat the sarcoma fluid with snake venom. And we're going to use a snake venom that chews up protein. And so if that stops the halo from forming, then it must be a protein, fully expecting that would happen. But thank goodness they had it controlled, because what they found was the snake venom alone was even better than the sarcoma at keeping the neurites alive and making them grow out. What? Wait a minute. The snake venom is itself having the same effect, and it turns out that was key for finding this stuff, because then what they later realized is that mouse submandibular glands also make this stuff in unbelievably large quantities. Actually, male mouse submandibular glands. And the same gland in snakes is being used to make this venom. So they eventually find stuff, isolate the stuff, and they name it nerve growth factor, or NGF. And this is before, molecular biology. This is in the late 50s, early 60s. So, nobody knows how to manipulate genes and stuff like that. So in these old-fashioned fractionation methods with old sort of brute force biochemistry, you'd get a whole bunch of stuff, and then there were various things you could do. You dissolve some of it in ethanol to see if it's ethanol soluble, for example, or you treat it with snake venom to see if it's a protein, stuff like that. And in those old-fashioned assays, old-fashioned methods, for every step you took to try to isolate a fraction, you always lost like 100-fold of whatever it is you're after. So it's very tedious, etc. So they were lucky in two ways. Number one, they picked the one sarcoma that made NGF. And number two, they picked a snake venom that was chock full of NGF too. And so it'll turn out that NGF is absolutely crucial for the survival of dorsal root ganglion cells and for sympathetic cells. So here's one way you can look at it, where you can keep track of how much NGF you provide in a dish. And the more NGF you provide, the more of these, in this case, sympathetic neurons, the ones that normally live in the sympathetic chain, the more of them that survive. Or in another set of experiments that were done by this time in the late 70s, early 80s, you can induce pregnant rats to make antibodies to NGF. And when you do that, their pups developing inside them have tiny little dorsal root ganglia, hardly any cells there, showing that it's antibodies to NGF that are, in fact, keeping them from living. And so with experiments like that, it became clear that normally every target in the body that is going to get sympathetic innervation or DRG innervation makes NGF and releases it. And once neurons innervate those targets, the number of neurons that survive is directly proportional to how much NGF that target is making. Everyone get the idea? And so you have this beautiful matching of the size of the periphery to the number of DRG neurons and the number of sympathetic neurons. So it's a nice way of matching things up. How's that so far? It turns out, and so NGF was one of the first substances that was said to be a neurotrophic factor with a pH. And the idea is it's feeding these neurons and keeping them alive. And if there isn't enough of the neurotropic factor out there, the neuron will starve and die. But it also turns out then NGF is neurotropic, that it attracts cells. And one of the most dramatic indications of that was that when people were keeping a dorsal root ganglia cell in culture, and then the dotted lines here show you a glass microelectrode. It's out of focus, but that's been loaded up with NGF, and then they could pass a current to let some of it out in a very nice, slow way. And so the growth cone in this dorsal root ganglia starts growing towards the pipette. And if you keep moving the pipette, you can get the growth cone to go in whatever direction you want, showing that it is following this NGF trail. So NGF is a neurotropic effect. It helps the neurons to reach the target in the first place. Hey, you're a sympathetic cell. You like NGF. I've got it for you right over here. Come over here and innervate here. And then after the innervation is established, then apparently there's not enough NGF for everybody. And depending on the system, there's just enough NGF to keep about half the neurons alive. And that means that those targets that need more sympathetic innervation produce more NGF and end up with more sympathetic innervation.
Why don't you start up with more neurons and what the third thing?
It's a great question. It seems wasteful, right? I mean, it's not free to make a new neuron, have it go through differentiation. And the thing that experiments where you put on an extra limb bud tells you, they're not doomed. That was one of the questions was, well, maybe you make too many, but some, it's like natural selection. Some just don't make. They made some mistake. But wait a minute. If you give them a bigger target and more neurotrophic factor, they stay. And apparently they work just as well as any other. So they were perfectly healthy. And so they're not mistakes, right? They did their job. They went through the steps of differentiation, but there aren't as many of them. And of course, as usual, I can't tell you why that's true, but I can tell you it's so universal across all the species and every part of the nervous system that for whatever reason, however expensive it must be, it's not expensive enough, i.e., animals keep doing it. Now, it's always possible that the reason that happens, sometimes natural selection, there's always so much you can work with, right? Once the species have gone down a certain pathway, it may be near impossible to have a change happen early in development to keep that from happening. So for example, don't tell me how perfect the human body is. Mammals breathe and eat through the same tube. That's dope. That's really a dumb idea, right? But apparently only a few species have found a way around. You think whales, right? Things like that. And there had to be a pretty compelling reason, natural selection, to take them in that direction. And so it may be the same case here, that we've been so busy making extra cells all along that it's just, too hard for natural selection to find a way to stop it. Just
C. elegans, there's no cell-to-cell communication, like if it's pre-slated to die, it'll just die.
In almost every case. So in C. elegans, they stick to that mosaic system of differentiation, right? Mosaic termination of cell fate.
Because I could always see how it would be beneficial in if you weren't predestined to die, like, I need a certain number of cascades.
There especially, you might think that if a mutation arose that kept that other cell from dying, stick around just long enough to see if you're needed. And again, we talked about this in terms of the history of transcription factors that these two cells have been exposed to. They've got to be pretty similar, right? Until that very last divide. And so you can imagine it might happen there, but it doesn't. And probably, so much of what makes C. elegans useful is the fact that you got to remember, I don't know how many trillions of them there are on the football field at MSU Stadium, right? Lots. They're just bare bone organisms, right? There's just all it is 1,000 cells, 300 neurons, and the only reason they're still with us is because they manage to reproduce. So, no more logical than that. So far, you all right? And notice I haven't said anything about motorons, and I want to preview now. NGF does nothing to keep motorons alive. So it's very specific. It works great for sympathetic neurons and dorsal root ganglion neurons, but it's not important for every neuronal population. And then there was a great idea that told us a lot about how NGF works. A guy named Campano took advantage of these little devices, these little pieces of metal that were sold for electron microscopy reasons. But he noticed that they were kind of cool because they had, two compartments. And so he had the idea of putting a little bit of Vaseline on one side and setting it down in a culture dish. Okay. And they were somewhat there was some diffusion out of these things. I can't remember now if they're, Teflon, inserts. And so he would grow dorsal root ganglion cells or sympathetic neurons in a dish here. And as long as there was NGF there, the neurons would grow and they'd stay alive. But what happens if he's growing them in a dish and the cell bodies are in the outer, the bigger compartment, and the neurites grow under the piece of Teflon and grow? And what if there's only NGF there? And what he found was that whichever compartment had NGF, that's where all the neurites would grow. And as long as there was NGF in one of the compartments, either in the compartment where the cell body was or in one of the compartments where the neurites were, the cells would stay alive. So the NGF could be taken up by neurites and would tell the cell body, don't die. We're getting our NGF. And why are there more neurites in this compartment? Because that's where the NGF is, because sympathetic cells are, programmed through differentiation to seek out NGF as a neurotropic factor. You all right? I'm going to stop telling a story and just skip to what we understand about what's happening here. NGF binds to a receptor that's known as track A. And when NGF binds to molecules of track A, the extracellular portion of it, it causes them to bind together and form a heterodimer, and that changes the intracellular portion of the track A receptor. So, and when that happens, the cell will then undergo pinocytosis and incorporate that whole thing. And then it gets attached to microtubule and heads to the cell body, and the NGF tells the nucleus back there, hey, you're getting neurotropic factor, don't die, stick around. And there are a whole bunch of other proteins that are important for this process, but they're not relevant for us. If you're an NGF aficionado, then that's where, lots of interesting things happen, but we can skip over all that. So as long as some of the neurites get NGF and have the receptor to respond to the NGF, it'll get shipped to the nucleus and tell it, don't die. How's that? So NGF was for years the only identified neurotrophic factor. And in a world where people are very concerned about how neurons sometimes die in various diseases, that was a big deal to know eventually, again, using mouse submandibular glands, people were able to get enough NGF to actually decode the protein structure of the stuff, long before anyone had molecular biology to find the sequence and therefore deduce what the protein series would be. They did it the old-fashioned way. Yeah.
Does the NGF, does it personally get sent to the nucleus, or just the signal that it's?
It's a great question. The NGF, people would do, you could put radio-labeled NGF in one of those compartments and it would end up in the nucleus.
Okay.
So it's getting transported.
Is it the receptor and the NGF?
They're both shipped downstream, yeah.
And then it doesn't, it constantly needs NGF, right? So new receptors have to be.
It's a great question. In vitro, these cells constantly need NGF. You take the NGF away, they die. In development, there's only a stage at which the neurons have to have NGF. And if the NGF disappears after that, they go on and live. So there's a phase, where they are NGF dependent. And if they get through that process, then later they are NGF independent. Maria?
So it's all having that NGF, you feel let's say in mutation in the receptor where the neurites, where the NGF that comes to the neurites cannot get sent down to the cell body, then it would still die.
The cell will die. Yeah. If there's no way, and if the NGF doesn't get to the nucleus, where, of course, it's going to alter gene expression. If it doesn't get there, the cell will die. These cells will die at that stage of development. And when you're growing them in a dish, they're always at that early stage of development. They never finish differentiating so that they can become NGF independent. And I won't talk about it a lot, but I got to tell you, by 1986, I mean, I was already well in the field. It was like the structure of NGF, it was understood the old-fashioned way in the late 60s. And everybody in neuroscience is saying, when are they going to give the Nobel Prize for discovering NGF, the only identified neurotrophic factor? And it's been nearly 20 years now. What are they waiting for? And finally, in 1986, they award it for NGF, and everyone's kind of shocked that they don't award it to Viktor Hamburger, who started the research and in whose lab it was done. They award it to his two postdoctoral fellows, Rita, Levi-Montalcini, and Stanley Cohen. And that's especially perplexing because the rules are that they never share it with more than 3 people. So everyone's saying, what? Why not? Why did, and indeed it was Hamburger and another student who first found the tumor that started the whole process off. And anyway, so everyone was kind of shocked. Now, the rules of the Nobel Prize Committee are that they won't release any of their notes until 50 years after the prizes are awarded. It's all a secret, the deliberation and all. So in 2036, we're just 12 years away, we'll get to find out what the notes say about why Hamburger was slighted. And by 1986, you half wonder if they were hoping Hamburger would die because he was older than the others, so that they wouldn't have to face any controversy. But it's like, oh my God, it's way overdue. We got to do something. And again, anyway, so I'm holding on. Tonight, you'll be alive, almost certainly. So I'm going to hold on to see what happens. So, I'll only be 82. So, that made me no older than Trump would be if he got elected again. Okay, no more politics. So, and to make it personal, in 1988, I was on a sabbatical leave at St. Louis, at WashU in St. Louis, and Hamburger, he used a cane, but he walked in every Thursday to have a brown bag lunch. And so, it was like, and Levi-Montalcini's memoir had just come out. And, and she's amazingly silent about why, Hamburger didn't share. No protest from her about why. So it was pretty hard. And then, poor dears, he lived to be 101. So, So, although both the Levi-Montalcini and Cohen outlived him, as you expect, they were syllabus from the beginning. There are 3 times when I'm going to record a lecture for class. One is the Thursday before the October break, which is next Thursday. The other is Election Day. And the third is the Tuesday before Thanksgiving, right? So do not come here next Thursday because I'm going to record the lecture. Pardon? This coming Thursday, yeah. I mean, 2 days from today.
There won't be cahoots.
And there won't be any cahoots. Okay, so I'll record the lecture. I haven't yet because I don't know how far I'm going to get today. I'll record the lecture. I'll probably finish chapter 7, and it will be on D2L tomorrow sometime. You watch it when it's good for you, right? And the idea is, I know some of you travel, so same with the Tuesday before Thanksgiving and Election Day. I don't know why it's not a national holiday as it should be, but it's not. And I mean, yes, I do know it's not a national holiday because then people who don't have a lot of money would find it easier to vote. That's why it's not a national holiday. Okay. I said no more politics. I was lying. Okay. It turned out that NGF, once molecular biology came along, it turned out that that they could use molecular biology to look for other genes in the genome that were similar to NGF. And it turned out it was part of a big family of neurotrophic factors. The second one that was discovered was brain-derived neurotrophic factor, BDNF. It was called that because in this case, they harvested the brains of lots of chicks and amassed enough to do old-fashioned biochemical fractionation, and they were studying it. And then when molecular biology came along, they said, oh, you know what? That BDNF stuff you've been isolating from chick brains, it's a relative of NGF. And in fact, several more were found, and they got named neurotrophins. So the first neurotrophin is NGF, and the second neurotrophin was BDNF. And then some folks found another one that got named, then they ran out of names and just started numbering them, NT-3. And then folks found neurotrophin 4, and in a little kind of funny hiccup, then some other people found neurotrophin 5, and oh, dang, it's the same as neurotrophin 4. So those guys who thought they had found a new neurotrophin, they just missed out, right? Didn't quite. And it survived the literature long enough, they get lumped together as NT4/5, okay? Interestingly, there are other vertebrates where there are more neurotrophins than these 4, right? So in some fishes, there's 6 different neurotrophins, but we will concern ourselves with those. So that's one family. The neurotrophins is one family of neurotrophic factors. There are other neurotrophic factors that are not part of the neurotrophin family. So ciliary neurotrophic factor is some stuff that was found that keeps ciliary ganglia neurons in chicks alive. And there's others that we'll talk about later, the glial cell line derived neurotrophic factor. It was, basically, they could derive it from a glial cell line, right? GDNF, and we'll just call it that from now on. So far, so good. And those are different neurotrophic factors than the neurotrophins. By the way, Stanley Cohen certainly did other things to deserve a Nobel Prize, because after he left Hamburger's lab, he started looking for other neurotrophic factors, and he's the one who discovered epidermal growth factor, and that was another whole family of trophic factors. It just didn't happen to be for neurons. Just
Is it family that because they're two the diverging different?
Great question. In these cases, once again, why are there 4 neurotrophins? There must have been at least 2 cases where a gene was duplicated and then they started to diverge in function.
Okay, is that for all families? Like whenever they take
In general, when we talk about a family of RTK receptors, right? In the course of evolution, there came to be more and more cases of that happening, and they got to be useful in more and more places. And now what they enable is more and more specific signaling between cells. So yes, exactly. And same with the EGF family. And what's the other? And transforming growth factor, right? The TGF beta, that too is related to the EGF family. So, and just as there's several neurotrophins, there are also several neurotrophin receptors. And so the first one that was identified was the one that is specific for NGF, and it's known as TrkA. And people doing cancer biology discovered a related factor and named it TrkB before anyone realized it also happens to bind neurotrophin 2, which is BDNF. Biology is not usually that kind to students who want to remember something. So it's an easy mnemonic, right? That TrkB binds to BDNF and TrkA binds the first neurotropic factor. And then sure enough, there's a TrkC, and it's the one that binds the third identified neurotrophin factor. So far, so good. But it turns out that TrkB is also the one that binds neurotrophin 4. So our luck ran out. If only there'd been a TrkD, then we'd be able to keep them all straight, but it's not that way. And also, interestingly, while NT3 binds TrkC very well, it also has some affinity for This is useful to know because this can give you some idea of which cells might be sensitive to which neurotrophic factor, depending on which tracks they express. In addition to that, there's a so-called low affinity receptor, known as a P75 receptor, that binds all of these factors. And we won't talk a lot about the P75 receptor. It's from a different family. And the fact that it's not very specific for the various neurotrophins tells you that it's probably not having very specific neurotrophin-specific action. One speculation is that cells sometimes make the P75 receptor just to kind of keep any neurotrophins that are in the area around. And by low affinity, in a neuroscience class, you know what that means already. What we mean is when they bind to the receptor, the extracellular portion, they don't stay very long, right? And they're compared to when they bind to a high affinity receptor, where they stay longer, as they need to do, to cause, for example, the two halves of the two track receptor subcomponents to bind together, to be activated, and then do whatever intracellular stuff it's going to do. Oh, and there's yet another. Oh, yeah, just to say that the track receptors, this is what it stands for. I don't need you to remember that. But it is a RTK family member. Question, yeah.
You say that it goes back to the differentiation itself into the cell individually.
Yeah, we're not showing that here, but it in some cases it does.
So not all the growth factors
are incorporated and sent to the nucleus. As far as I know, they all are.
So what's the point of the law thing you want to say?
Yeah, it is not internalized, and so what is the point? We'll look at that. There is some effect in the postsynaptic output in the intracellular, in the target cell. There are some intracellular changes that happen, but they're not as much related to questions like living or dying.
Okay.
So it's just another one of those instances where natural selection has provided several ways to detect these things and several ways to respond to them. I've got a figure that tells you a little bit about P75 receptor in a bit. So the idea is that there are different neurotrophic factors that different neurons are sensitive to, and that not all are the same. So dorsal root ganglion cells, for example, love NGF, and they also respond to BDNF, and they also respond to NT-3. So they're somewhat responsive to all those. We can take a peripheral ganglia, the nodose ganglia, for example. It doesn't care about NGF at all. It has some response to BDNF, but it's really crazy about NT-3. Or we can look at sympathetic ganglia that love NGF, seem totally indifferent to BDNF, and barely respond to NT-3. So you get the idea that with different neurotrophic factors, different neurons that are sensitive to different factors, that this is a way to match innervation of the number of neurons to various targets so that you have the right kinds of neurons innervating sympathetic targets, for example, the right sort of neurons innervating dorsal root ganglion targets. And remember, the dorsal root ganglia, these are the cells, their cell bodies live in the dorsal root ganglia, but they're going to send their axons out to the periphery, and they're going to provide all the sensory information from the body, right? Pain, touch, stuff like that. And here's a little cartoon. I don't need you to memorize this, just pointing out that various parts of the periphery are sensitive to different neurotrophic factors. And this is how you can end up with this sort of matching of the size and numbers of each. Once people started knocking out track receptors, they could see, comparing in mice, you knock out the track A receptors and what percentage of DRGs survive, right? What percentage are lost, right? So this is neuronal loss. Like 78% of them, the superior cervical ganglia, the topmost sympathetic ganglia, almost all the cells die. The trigeminal ganglia, they're in the periphery. Most of the cells die. Other ganglia pay no attention. Track B knockout mice, on the other hand, they don't lose any ganglion sympathetic cells. They lose a lot of DRGs, and they lose some of the others, Track C, et cetera. Now, again, I don't want you to memorize these numbers, right? I just want you to know that different cell populations respond to different neurotropic factors. I absolutely want you to know that NGF is important for DRG cells and sympathetic cells. But beyond that, I just want you to know that it varies. So here's the picture I wanted to see, where this is a great simplification of track receptors generally responding to whatever their ligand is, the sort of pathways that people talk about in biochemistry that can alter the biochemistry of the target cell. And so, again, I don't want you to memorize these, not for my sake. I just want you to know that depending on the conditions inside the cell, activation can help cell survival or neurite outgrowth or other sorts of plasticity. And that the P75 receptor can also contribute to these things through quite separate pathways. Which particular pathway is going to get activated in which particular cell depends on which receptors it's expressing and depends on the internal conditions of the cell when the binding takes place. And all of these pathways are familiar to you, right? I mean, for those of you that are taking biochemistry, but I don't need you to remember any of them because all I care about is that they affect the cell. And it would turn out to be, once again, C. elegans, this incredibly simple animal, that would, in many ways, make it possible for us to have a better understanding of apoptosis in vertebrates. So early on, when they started making mutant C. elegans, they would occasionally come up with mutants where the cells would not die. Cells that they knew were normally programmed to die because of their mitotic lineage didn't die. And the convention among the worm people was to come up with names like this, where they call them cell death mutants, or SED. And they found several different mutations of different genes that could stop cell death from happening. And for the most part, what would happen is there'd be that last cell division. One daughter cell would take on a particular fate, and the other cell that normally would die would just sit there, looking pretty, undifferentiated, would just sit there and be there the rest of the animal's life, which, not very long, but, right? So some of these were very specific. And eventually they were able to track down the mechanisms behind it. So SED1 turned out to be a membrane protein that signaled to the immune system, come eat me up. I have no purpose here. Come gobble me up, a phagocyte, and let's make use of this stuff that I mean for something else. SED2 is a gene that's active in the phagocytes that causes them to devour a cell that's no longer around. And then SED3 was in some ways the most fascinating of them all. It was coded for a protease, an enzyme that chews up proteins. And it turns out that it's a cysteine-dependent aspartate-directed protease, and so they gave it the name caspase. And if you think about it for a moment, if you're a cell and you're trying to stay alive, transcribing and translating a bunch of pretty non-specific protein-dissolving enzymes is maybe not a good idea, right? So in fact, this protease is made specifically to start taking the cell apart from the inside. So, and yeah, how's that so far? Questions about that? Sam?
So currently I've covered that idea on input receptors.
Yes.
It is to presumably genes.
Yeah.
But we've also seen that NGF's buying cartitase
the track receptor is an RTK receptor. Yeah.
Yeah. So, and then you also said that every time then you have to find something that something will be translocated to make this. So is that RTK?
I wouldn't go that far, but if you remember back to that other figure, we showed that one figure where indeed that part of the membrane where it got bound is incorporated, is internalized, and then attached to the microtubule. And yeah, the whole assembly gets set down, including the track receptors.
And stuff.
Yeah. But I mean, they're still embedded in the membrane, right? But the membrane's this little bubble, this vesicle that's, yeah.
I have a question on this slide before. So,
Before this? No.
so what are you talking about? Like, I see points about our maybe through the eyes to this stuff.
Any track receptor.
No, there is all the kinds of I'm the missing.
So it depends on which of the pathways you're following, right? So for that leftmost pathway, eventually it gets translocated to the nucleus and affects gene expression. But, and then to remember that it'll be one part of the neuron's life where it is NGF dependent. But once it grows up, cell survival is no longer on the table, right? Once it's gotten past that stage where it was NGF dependent, now it's NGF independent and something else is going on.
But it's that say like these stuff need to be angial. Like so as if you don't
The other neurotrophins also, depending on which cell and which neurotrophins, there are times when they too are internalized and they all affect gene expression. All those pathways eventually affect gene expression. Just in more details than we need to talk about. And so the picture that I want you to get is that there are lots of different, pathways that can be incorporated this way. And you get this idea of that during development, populations of neurons are dependent on neurotrophic factors only temporarily, just long enough to decide how many is going to live and how many is going to die. Just as the period of apoptosis only happens early in development. So an interesting question is, what happens inside a cell that used to be totally dependent on NGF to survive so that now it no longer needs it? Well, the rough answer is the cells change, right? It has a different developmental history and a different history of transcription factors and gene expression. So it's a different kind of cell now. It's our little cell. It's grown up. Try not to shed a tear. Okay, so look for the next lecture on D2L and have a good break.
And that's